Alright, so today we're going to take a look at DNA replication and we're going to draw it out. And first things, we're going to have to know a few important details before we draw it out. The first thing is that DNA strands are anti-parallel. And what that means is we're going to have two strands here. And this top one is going to be going in a 5' to 3' direction. And this bottom one is going to be going in a 3' to 5' direction. So they're parallel, but they are anti-parallel because they're running in opposite directions. The second thing is that DNA has complementary base pairing. And what that means is you're going to have, we have 4 types of nucleotides that are found on DNA. We're going to have adenine that pairs up with thymine, and we're going to have guanine who pairs up with cytosine. So what this means is if you have a DNA strand and it is, let's say, adenine, guanine, cytosine, thymine, guanine, adenine, thymine. Because they're complementary, that means adenine is going to have to pair up with the thymine, and guanine is going to have to pair up with the cytosine. You can't have a adenine and guanine pair up or a thymine and guanine. It's going to be adenine and thymine always bonding with each other, and guanine and cytosine bonding with each other. So here we would have a guanine, here we have a thymine, so we're going to have an adenine, and so on. So it's going to look something like that. And the next thing we are going to take a look at is that the hydrogen bonds between the hydrogen bonds are going to hold nucleotides together. So what this is getting at is let's draw out a DNA. So we're going to have two strands that run anti-parallel, which means one strand is going to run in a 5' to 3' direction, and the other strand is going to run in a 3' to 5' direction. And this DNA is going to have these bonds that are going to hold the two strands together. And these bonds are going to be hydrogen bonds. And these hydrogen bonds ultimately decide how strong a DNA strand will be, because adenine and thymine only have 2 hydrogen bonds between them, whereas guanine and cytosine have 3 hydrogen bonds. So the more guanines and cytosines you have in your DNA molecule, that means it'll be stronger than if it were to have more adenine and thymine nucleotides bonding together, because of the simple fact that guanine has 3 hydrogen bonds, whereas adenine and thymine have 2 hydrogen bonds between each other. And our last thing that we're going to take a look at is that DNA is semi-conservative. And what this essentially means is that we start off with 2 parent strands. We'll call them parent strands. These are the original DNA that is going to be replicated. It's going to turn into 2 new sets of DNA molecules. We have one here and one here. And that means a total of 4 strands, because each DNA molecule has 2 strands. So if we have 2 DNA molecules, we're going to have 4 strands. So in this case, one of these parent strands goes over here, and the other one's going to go over here. And what that does is allow for 2 new DNA molecules, and you're going to have 2 daughter strands, and these daughter strands are going to run anti-parallel to the parent strand. So if this is our 5' end, this is going to be our 3' end, and this will be our 5' end over here, and this would be our 3' end over here. And same thing with this one up here. So now we can actually go and try our replication. So what we need to know first is that topoisomerase is an enzyme who is going to come and it's going to unwind the DNA. It's going to start here in these areas where the DNA is coiled up, like right here. Because DNA is not you can't really see it here because it's two-dimensional, but DNA is twisted and it's clumped together as you would see right here. See how it's that double helix formation that we learn about in our biology class. It's twisted, and it would just be a pain in the butt for DNA replication to go on if it's like this. So what topoisomerase does is he unwinds that DNA so that way it can look like this two-dimensional shape over here. Nice and easy to work with. And the next thing that is going to come in is we're going to have our helicase. And the way I like to think about helicase is that it's going to come in and break the hydrogen bonds between the two DNA strands. And you can think of helicase as a helicopter blade, because helicopter blades are sharp so they can cut. So you can think of helicase coming and just cutting down all these hydrogen bonds in the way in order to start replication, or in order to have the DNA strand prepared for replication to begin. So the third thing we're going to take a look at is DNA polymerase, or DNA primase will lay down a primer in order to begin replication. So what that's going to look like is we're going to have a little primase, DNA primase, and it's going to lay down this RNA primer right here. And that primer is going to be it's going to be made with RNA nucleotides, and it's going to be the point where DNA polymerase comes in the next step and it's laid down. So what the primer does is say, hey, DNA polymerase, come over here and start working right here. So what DNA polymerase will see that and immediately hop on right here. So which takes us to our next step. It's going to, DNA polymerase 3 will add the nucleotides to the parent strand in a 5 to 3' direction. So what this means is, as this guy is going forward, he's adding nucleotides. And what those nucleotides are going to look like are it's just going to be another strand. So as he moves forward, he's going to be creating more and more nucleotides. And this can only, DNA polymerase can only do this in a 5' to 3' direction. So what does that mean for the other strand? Well, because the other strand it goes from 3 to 5', that means it's going to have to work backwards, because it cannot go from 3' to 5' direction. DNA polymerase can only add nucleotides in a 5 to 3' direction. So what this is going to do is it's going to call for a primer, and so DNA primase will come lay down your primer right there, and then you're going to get another DNA polymerase. Oops, that's not a circle. You're going to get another DNA polymerase come down, and it's going to start right here. And because it can only go in a 5 to 3' direction, it won't be going this way. It can only go this direction. So you'll have a new strand of DNA that looks, after that is done, it's going to look like this. And as this is going to have to continue, so it's going to have to call for another primer That primer will allow that DNA polymerase to continue to bind to it, which will allow for more DNA to be synthesized through DNA polymerase 3. And it's going to go and do that all the way till it reaches back here. And that's as topoisomerase unwinds and helicase continues to break these hydrogen bonds. So what do we need to know about a leading and lagging strand? Well, because this strand here is having to work backwards, and it's having to come all the way over here and then back this way and then over here, this is going to happen a lot slower than the strand up here on the top, which means this is going to be our leading strand because it's happening faster. And this right here is going to be our lagging strand. And that's simply because of the basic concept that DNA polymerase 3 adds nucleotides only in the 5' to 3' direction. So because it adds it only in the 5' to 3' direction, and because this because DNA molecules are anti-parallel, that means that this strand is going to have to go in this direction, which means as this breaks more bonds, it's going to have to start right here, move along. And then as soon as it does that, by the time helicase is over here, it'll have to jump back over here and then start there. And it's going to take a lot longer than the leading strand. So the next thing we need to know is that DNA polymerase 1 is going to come and it's going to remove these primers that were left, because these primers took space for DNA, where DNA could be. So these primers are going to have to be filled in with little gaps of DNA, and that's what DNA primase is going to do. It's going to fill those little gaps right there with more DNA that's complementary to the bases of the parent strand. And it's going to do that for all these little areas that there was a primer in order to conserve genetic variation in our genetic code. So it's going to go through the entire DNA molecule to every primer that was laid down, and it's going to fix and add nucleotides to all those areas. Now that leaves us with our last thing. We're going to have something called a DNA ligase that's going to come, and you can think of it as a little worker with duct tape. It's going to come here with his arms, and it's going to seal these patches of DNA that were not connected because the Okazaki fragments were made separately, especially on the lagging strand. They were made separately because of that 5' to 3' direction that it could only occur in. So it's going to seal all these little areas right here, and you're going to get that down the entire molecule over here as helicase breaks more and DNA polymerase makes more nucleotides. So you're going to get him over here to where there were other fragments, and ultimately it's going to end with a total of 2 DNA molecules. So you'll get 2 DNA molecules, one that has the parent strand, or 2 that have one parent strand of each. So we call the blue one the parent strand. The red one is going to have to be the daughter strand. So we're going to have 2 parent strands and then 1 daughter strand.